Disclaimer: I do not own the rights to the images or information. This was strictly made for the purpose of studying for boards. The neural tube, the central nervous system, CNS, is a tubular structure originating from a proliferation of ectodermal epithelial cells referred to as the neuroectoderm, which is located dorsal to the notochord along the axis of the embryo. This thickened ectoderm, known as the neural plate, invaginates along this axis, forming a groove until the lateral extremities of the original plate, the neural folds, meet centrally and fuse over the neural groove to form a neural tube and canal. As the neural tube forms, it separates from the non-neural ectoderm, which grows over the dorsum of the tube to fuse along the midline. As this fusion and separation of ectodermal layers occurs, a longitudinal column of ectodermal epithelial cells arises from the junction of non-neural and neural ectoderm and separates from these two structures when the neural tube is formed. These two bilateral columns, situated dorsolateral to the neural tube throughout its length, are the columns of neural crest cells. Figure 3-1. Closure of the neural tube progresses rostrally and caudally from the level of the site of development of the rhombencephalon, which is the most caudal division of the brain. The caudal closure forms the majority of the spinal cord. Closure of the brain portion of the neural tube may initially occur at multiple sites and progress rostrally and caudally. The locations of these sites vary among species of animals. Prior to complete closure, the most rostral opening is the rostral neuropore. Figure 3-2. The caudal portion of the spinal cord develops from the caudal end of the closed neural tube as an extension of a column of neuroepithelial cells that grows caudally on the midline between the notochord and skin ectoderm. A cavitation of this column of cells produces an extension of the neural tube and its neural canal. This portion of the neural tube will ultimately form the caudal, the sacral, and a variable number of lumbar spinal cord segments. An opening may persist at the caudal end of the neural tube, allowing communication with the subarachnoid space of the leptomeninges at the conus medullaris. The rostral end of the neural tube develops rapidly and produces three vesicles, from rostral to caudal: the prosencephalon, the mesencephalon, and the rhombencephalon. Figure 3-3. Early in its development, the prosencephalon has lateral enlargements, the optic vesicles, which grow later ally to contact the overlying skin ectoderm. The further development of this primordial eye is described in Chapter 14, Visual System. Two additional swellings emerge from the rostral prosencephalon and grow out of the neural tube on each side laterally and dorsally. These telencephalic vesicles completely overgrow the original vesicular system and form the cerebral hemispheres. The portion of the prosencephalon that remains at the rostral end of the neural tube is the diencephalon. The optic vesicles remain associated with the diencephalon. The neural canal within the diencephalon is the third ventricle. It communicates rostrolaterally with the neural canal of each telencephalon, cerebral hemisphere, which are the lateral ventricles, first and second ventricles. The small communications on each side connecting the lateral ventricles with the third ventricle are the interventricular foramina. The nuclei of the thalamus and hypothalamus develop in the diencephalon. The neurohypophysis is a ventral outgrowth of the diencephalon. The cerebral cortex and majority of basal nuclei develop in the telencephalon. The neural canal of the mesencephalon is reduced to a narrow tubular space called the mesencephalic aqueduct. From the rostral rhombencephalon, the cerebellum, or dorsal metencephalon, develops dorsally. The remaining ventral metencephalon becomes the pons. The caudal rhombencephalon forms the myelencephalon, or the medulla oblongata. The fourth ventricle is the lumen of the neural canal in the rhombencephalon. It communicates with the meningeal spaces that develop around the neural tube by way of openings that arise in the wall of the neural tube caudal to the developing cerebellum. These openings are called the lateral apertures. See figures 4-2 and 4-4. The neural canal continues caudally as the central canal of the medulla spinalis, the spinal cord. Cell differentiation in the first stage of development within the wall of the neural tube. The cells, which are commonly referred to as neuroepithelial or neuroectodermal cells, are organized in a pseudostratified arrangement. Thus, the neural tube is one cell in thickness. The cell membrane of each cell spans the full width of the neural tube, but the nuclei are located at various levels within each cell. These cells are all methodically active, increasing the thickness of the wall of the tube. The nuclei migrate within the cytoplasm of each cell, and their position is dependent on the cell stage of mitosis. During interphase, the nuclei are located on the external surface of the neural tube. Chromosomal deoxyribonucleic acid, DNA, duplication occurs with the nucleus in that position. As the nucleus enters mitosis, it migrates through the cytoplasm to the luminal surface of the neural canal. The peripheral portion of the cytoplasm in the cell membrane also retract to the luminal position, where cell division is completed. The two new daughter cells extend their cytoplasm and cell membranes back to the external surface of the neural tube. Their nuclei migrate back to the periphery again. In this position, these cells may undergo another mitosis or may differentiate. Because the nucleus is at the external surface during interphase, differentiation occurs at the external surface of the neural tube. Thus, in a short time, a new layer of differentiating cells appears on the external surface of the actively dividing layer. The mitotic layer of cells is the germinal layer. The cells undergoing differentiation form the mantral layer. Figure 3-4. The cells that are differentiated are of two types. 1. Immature neurons and 2. Spongioblasts. Immature neurons are the primary parenchymal cells of the nervous system. They are often referred to as neuroblasts, but this is a misnomer because once a neuron is formed, it will not divide again, as this term implies. The differentiated immature neuron grows extensively, forming long processes and becoming a mature functioning cell. However, with a few exceptions, it does not divide again. Spongioblasts are the progenitors of the neuroectodermal supporting cells of the nervous system, the neuroglia, GLU. Two of the three forms of glial cells, astrocytes and oligodendrocytes, are derived from these spongioblasts. See Figure 3-4. The third glial cell is the microglial cell, which is mesodermal in origin. It is a monocyte that enters the nervous system from the blood supply. As the primitive neurons and spongioblasts are differentiated and grow and the neurons produce processes, the neural tube becomes arranged in three concentric layers. Figure 3-5. Adjacent to the lumen of the neural tube is the germinal layer of proliferating neuroepithelial cells. This proliferative mitotic activity will ultimately be exhausted, reducing the germinal layer to a single layer of cells that range from squamous to columnar and are called ependymal cells. These ependymal cells line the entire lumen of the neural tube, which includes the ventricular system in the brain and the central canal of the spinal cord. Peripheral to this germinal layer in the embryonic neural tube is the thick layer of differentiated cells, immature neurons, and spongioblasts. They form the mantral layer, which will ultimately become the gray matter of the definitive spinal cord, the nuclei of the brainstem, the nuclei and cortex of the cerebellum, and the basal nuclei and cerebral cortex of the cerebrum. This three-layered relationship persists in the developing spinal cord. In the telencephalon, these neurons migrate from the mantral layer to the external surface of the neural tube. The initial external layer of the neural tube is the marginal layer. It is composed primarily of the growing axonal processes of the neuronal cell bodies in the mantral layer. These axons will be myelinated by the oligodendroglial cells, forming tracts in the white matter. From the mesencephalon caudally, a longitudinal groove, the sulcus limitans, appears in the lateral wall of the neural canal. Thus, the neural canal can be artificially divided into dorsal and ventral portions by an imaginary dorsal plane at the level of this sulcus. The dorsal portion is called the alar plate and the ventral portion the basal plate. Functionally, the alar plate mantral layer is concerned predominantly with sensory systems and the basal plate mantral layer with motor systems. See Figure 3-5. Neurogenesis. Having established the role of the neural tube in the initiation of the development of the CNS, it is appropriate to consider neurogenesis in the adult animal. It is recognized that once a primitive neuron is differentiated in the neural tube mantral layer, that cell will not undergo mitosis again and the mature neuron will persist for the life of the animal. The premise is that once the CNS is formed, neurogenesis ceases to occur. If a population of neurons is lost because of disease or injury, those neurons are not replaced. Although a finite time in development exists for most neurogenesis, substantial research supports selective postnatal neurogenesis throughout the life of the animal. In the embryonic neural tube, the embryonic neuroepithelial cells give rise to a cell line referred to as radioglia. During development, these radioglia function to guide the developing primitive neurons to their sites of destination in the neural tube, such as the cerebral cortex. However, in addition, it is now recognized that these radioglia may also differentiate into primitive neurons, astrocytes, oligodendrocytes, and ependymal cells. After development is completed, the undifferentiated radioglial cells populate the subventricular zone, which is most evident rostrally in association with the lateral ventricle of the frontal lobe. Here, these radial astrocytes function as neuronal stem cells as well as a source of astrocytes and oligodendrocytes. This adult neurogenesis occurs primarily in the rostral cerebral subventricular zone, where the newly formed neurons primarily populate the olfactory bulb. The other site is the subgranular zone of the hippocampal dentate gyrus. where they serve to replace dentate gyrus granule neurons. The activated neuronal stem cells at these sites are derived from a unique population of radial glia that is adjacent to the ependymal cells at these sites. The function of these cells formed in adult neurogenesis both in the subventricular and subgranular zones needs further study with regard to their purpose in mammals. These subventricular zone adult stem cells are thought to be the source of some of the gliomas that occur in the brains of humans and domestic animals. It is also thought that these cells may play some role in plasticity after injury. Additionally, these cells, it is hoped, may be harvested for cell replacement after devastating injury to the CNS or neurodegenerative processes. Medulla spinalis, spinal cord. In this text, the term spinal cord is used, rather than medulla spinalis, the nomenclature preferred by Nomina Anatomica Veterinaria. The development of the nervous system is dependent on complex genetic factors. Two signaling centers have been recognized as being responsible for the development of the spinal cord portion of the neural tube. One is the surface ectoderm adjacent to the ectoderm that formed the neural plate. These surface ectodermal cells secrete bone morphogenetic protein 4, BMP4, a cytokine that enters the neural tube and directs the development of the dorsal portion of the neural tube. BMP4 is responsible for triggering the secretion of transforming growth factor beta, TGF Greek letter beta, which diffuses ventrally in the neural tube, where it is responsible for the formation of dorsal horn neurons. The second signaling center is the notochord and its secretory signaling molecule, sonic hedgehog, SHH. SHH diffuses dorsally in the neural tube and is responsible for directing the development of the ventral portion of the neural tube with its ventral horn neurons. The spinal cord provides the least complicated example of the symmetric development of the neural tube by layers, ventral growth of the two basal layers and associated marginal layers beyond the level of the floor plate. Figure 3-6 leaves a separation between the two sides, which is the ventral median fissure. The mantral and marginal layers of the alar plates grow dorsally. The dorsal marginal layers fuse on the median plane to form a dorsal median septum that may be poorly defined. The external margin of this septum forms the dorsal median sulcus. This midline growth displaces the roof plate region ventrally, resulting in a reduction of the neural canal to form the small central canal of the spinal cord lined by ependymal cells. The mantral layer of the alar plate becomes the dorsal gray horn, also referred to as column, and that of the basal plate becomes the ventral gray horn. The mantle zone at the plane of the sulcus limitans becomes the lateral gray horn. See figure 3-6. Not only does a gross topographic differentiation of function of primitive neurons exist between the alar and basal plates, but neurons are further arranged in functional columns within the mantle layer of each plate. The general visceral afferent and general visceral efferent neurons are located adjacent to each other in their respective gray columns on either side of the dorsal plane through the sulcus limitans. The general somatic afferent, XA, and general proprioceptive neuronal columns are located dorsally in the alar plate of the mantral layer, and the general somatic efferent, XA, column is located ventrally in the basal plate of the mantral layer. Because the relative size of the components of each spinal cord segment depends on the volume of tissue being innervated, at the levels of the limbs the spinal cord segments responsible for their innervation are enlarged, forming the cervical and lumbosacral intumescences. The ultimate growth to maturity of a neuron in the peripheral nervous system depends on its appropriate innervation of a muscle cell, XA, or formation of a peripheral receptor, XA, general proprioception, GP. The lack of such innervation results in the degeneration of the neuron. In the cervical and thoracolumbar regions where appendages are not innervated, the immature primitive neurons in the basal plate mantral layer and the adjacent spinal ganglion that fail to innervate structures will degenerate by a process of cell death referred to as apoptosis. The shape of the ventral gray horn depicts the results of this process. In the basal plate mantral layer, the XA neurons located medially innervate the axial skeletal muscles. Those located laterally innervate the appendicular skeletal muscles. Within these areas of the ventral gray horn, the XA neuronal cell bodies may be further grouped according to the specific nerve that contains their axon and by the specific muscles they innervate. The growth of axons of the basal plate neurons through the marginal layer and outside the neural tube forms the ventral root and part of the spinal nerve and further branching of the nerves as they extend to the muscles that they innervate. This includes the general somatic efferent neurons located in the ventral gray horn and the general visceral efferent neurons located in the lateral gray horn adjacent to the sulcus limitans. These latter general visceral efferent, GV, neurons are the preganglionic lower motor neurons of the autonomic nervous system. This lateral gray column is only present in the thoracic, cranial lumbar, and sacral spinal cord segments. In the other segments, it was present in the embryo but subsequently degenerated because of the absence of a target organ or biochemical attractant. These GVE axons terminating ganglia that contain cell bodies of the postganglionic axons in this 2-neuron lower motor neuron system in the peripheral nervous system. Figures 3-7 and 3-8, see also figure 3-6. Neural crest at the junction of neural plate and surface ectoderm. BMP and WNT signaling proteins induce the ectodermal cells to develop mesenchymal features, penetrate the basal lamina, and form a column of cells dorsolateral to the neural tube. This bilateral column extends the full length of the neural tube and is referred to as the neural crest. These neural crest cells located along the developing spinal cord segments provide the neurons that form the spinal ganglia at each segment. Adjacent to each somite, proliferation of neural crest cells forms the segmental spinal ganglion. See figures 3-1 and 3-7. One portion of the axon that emerges from each of these cell bodies grows centrally into the spinal cord segment to enter the alar plate dorsal gray horn, forming the dorsal root. The other portion of the axon grows distally to form a sensory component of the spinal nerve and its further branches. The point of penetration of the marginal white matter layer of the spinal cord segment by the axons in the dorsal and ventral roots divides the spinal cord white matter processes into 3 regions called funiculi. These are the dorsal, lateral, and ventral funiculi on each side of the spinal cord. The formation of spinal ganglia is only one of many outcomes of neural crest differentiation. Prior to its segregation into spinal ganglia, an early migration of cells from this neural crest column provides melanoblasts to the somatic dermatome and adjacent epidermis and to the cell bodies of postganglionic axons in the 2-neuron GVE system. These latter cell bodies form the ganglia of the sympathetic trunk and the abdominal plexus sympathetic ganglia, as well as the medullary cells in the adrenal gland. See figure 3-8. These adrenal medullary cells do not grow any processes but synthesize and elaborate into the bloodstream the same endocrine substance, norepinephrine, that is the neurotransmitter released at the telodendron of the sympathetic postganglionic axon derived from the neural crest cells. Although the melanoblasts and GVE neurons seem unrelated, their common denominator is the unique metabolism of tyrosine, which provides melanin for the melanocytes and norepinephrine for the neuron. In addition, extensive migration of the neural crest cells occurs into the wall of the developing gastrointestinal tract. These will form the ganglionic neurons of the parasympathetic portion of the GVE lower motor neuron as well as interneurons, and they form the glial cells that develop in the wall of the bowel, creating what is referred to as the enteric nervous system. The latter is extremely extensive and complex and presumably is entirely derived from the neural crest cells. A similar migration forms the ganglionic parasympathetic neurons for the urogenital system. In addition to these nervous system structures, the neural crest contributes to the formation of bone and cartilage in the skull and derivatives of the branchial arches, to the wall of the great vessels at the base of the heart, and to thyroid parafollicular cells, odontoblasts, a portion of the leptomeninges and the lemocytes, Schwann cells, that form the myelin of the peripheral nervous system. This is a remarkable display of developmental capabilities in an initial column of cells. When any doubt exists about the origin of a structure, it is worth considering the neural crest for obtaining answers. Many studies have been and are continuing to contribute to our understanding of the molecular basis for brain development. Signals derived from homeobox, HOX, genes are expressed in the notochord, the precordial plate, and the rostral neural plate, which influence the regional specialization of the brain into its prosencephalic, mesencephalic, and rhombencephalic divisions. As in the development of the spinal cord, the signaling protein sonic hedgehog, SHH, secreted by the precordial plate and the rostral notochord influences the ventral patterning of the brain, and BMP4 and BMP7 secreted by the adjacent surface ectoderm control the dorsal patterning of the brain. Myelencephalon, medulla oblongata. In this text, the term medulla oblongata is shortened to medulla. The medulla is the most caudal portion of the brainstem and is continuous caudally with the spinal cord. The basic formation of the medulla involves only a slight modification of the development described for the spinal cord. The narrow dorsal roof plate of the initial neural tube, see figures 3-5 and 3-6, is stretched extensively instead of being obliterated by the proliferating alar plate and marginal tissue as it is in the spinal cord. Imagine grasping the midline roof plate of the neural tube with both hands and then pulling your hands apart, sideways. This would stretch out a thin layer of neural tube, roof plate, and displace the entire alar and basal plates to a lateral and ventral position. Figure 3-9. This would enlarge the lumen of the neural tube to form the fourth ventricle of the medulla, which is covered dorsally by only a single cell layer of neuroepithelial cells. At this site, These cells will not enter mitosis but will remain as a single layer of ependymal cells. The sulcus limitans that is present on the ventrolateral wall of the fourth ventricle provides the plane of division of the medulla into a ventromedial basal plate and a dorsolateral alar plate, which have the same functional significances in the development of the spinal cord. See Figure 3-9. Throughout the brainstem, the mantral layer of the neural tube is broken up into nuclei that are collections of neuronal cell bodies with a common purpose, and they are interspersed with neuronal processes. Some nuclei are more distinct than others. The described functional columns in the spinal cord have a similar location in the brainstem. In addition, the neurons in the medulla are organized into functional groups that are present only in cranial nerves. See Figure 3-9. In domestic animals, cranial nerves VI through ZI and the trapezoid body are part of the medulla. The rostral border of the medulla is the caudal border of the pontine transverse fibers. In primate and some non-domesticated species, the transverse fibers of the pons expand caudally to cover the trapezoid body, so cranial nerves VI through VIE are included with E in the pons. Cranial nerves VI, IX, X, XI, and ZI contain general somatic efferent neurons. The medullary nuclei of cranial nerves VI and ZI are located in an interrupted column along the median plane adjacent to the fourth ventricle. The hypoglossal nucleus is very long. The xenerinal cell bodies of the facial nerve migrate from their initial formation in the mantral layer to form the facial nucleus in a ventrolateral position, which is closer to their common source of sensory stimuli coursing into the medulla in the spinal tract of the trigeminal nerve. This phenomenon of migration is referred to as neurobiotaxis. As a result of this migration, the axons leaving this facial nucleus initially course dorsomedially to the floor of the fourth ventricle before turning to course ventrolaterally to leave the medulla and form the facial nerve. The XS cell bodies of cranial nerves X, and XI undergo a similar migration ventrolaterally and accumulate in the nucleus ambiguous, which is aptly named because it is a poorly defined nucleus. The preganglionic neurons of the parasympathetic portion of the GVE system are located in an interrupted column just ventromedial to the sulcus limitans, similar to their local chine in the spinal cord, where they comprise the lateral gray horn of cell bodies. Their axons leave the medulla when cranial nerves VI, IX, X, and XI. The sensory components of cranial nerves associated with the medulla arise primarily from primitive neurons that develop from the column of neural crest cells, with a few arising from ectodermal cells that proliferate from branchial arch ectoderm. The latter areas are referred to as cranial placodes. Neural crest cells form the sensory ganglia of cranial nerves VI, IX, and X, which are associated with general visceral afferent and special visceral afferent, taste, function. Ectodermal cells derived from the otic placode form the sensory ganglion of cranial nerve VI, which is associated with special proprioception for vestibular system function and with the special somatic afferent system for auditory function. These cranial nerve VI ganglia are located in the inner ear within the petrosal portion of the temporal bone. Their axons course into the alar plate region of the medulla to synapse on cell bodies comparable with the dorsal gray column cell bodies in the spinal cord. See Figure 3-9. The leptomeninges that surround the entire developing central nervous system, CNS, neural tube, arise from neural crest cells and adjacent mesodermal cells. These meninges contain the bulk of the blood vessels that supply the CNS and the roots of cranial and spinal nerves. The roof plate of ependymal cells, which covers the fourth ventricle and the thin layer of vascularized pia associated with it, is called the telechoroidea of the fourth ventricle. The capillary blood vessels in this telechoroidea proliferate to form the two longitudinal rows of a dense capillary bed. The adjacent ependymal cells enlarge into cuboidal cells, and the entire structure, cuboidal ependymal cells, pia mater, and capillary bed, protrudes into the lumen of the fourth ventricle. Figure 3-10. This is called the choroid plexus of the fourth ventricle. By strict definition, only the proliferated capillary bed is the plexus. Thus, the choroid plexus of the fourth ventricle comprises two sagittal lines parallel to and on either side of the median plane. These extend rostrally from the caudal part of the fourth ventricle to the level of the cerebellar peduncles, where each plexus turns laterally. At this point, an opening, called the lateral aperture, develops in the medullary roof plate. This aperture allows communication between the lumen of the fourth ventricle and the subarachnoid space that develops in the leptomeninges. At the level of this lateral aperture, the choroid plexus protrudes from the lumen of the fourth ventricle out through the aperture, where it is visible on each side at the cerebellomedullary angle. See Figure 3-10. The aperture itself is invisible grossly because it is filled with this choroid plexus. The choroid plexus is a major site of formation of cerebrospinal fluid, CSF. See Chapter 4. In domestic animals, The lateral aperture is the only communication between the ventricular system of the brain and the subarachnoid space, which makes it critical for the maintenance of normal intracranial pressure. ICP. Primates have an additional median aperture located on the median plane and caudally in the caudal medullary velum of the fourth ventricle. Metencephalon, cerebellum and pons. The initial development of the metencephalon is comparable with that of the myelencephalon. Cranial nerve V, the trigeminal nerve, is associated with this segment of the brainstem. Figure 3-11. Its motor neurons arise in the basal plate mantral layer, migrate a short way ventrolaterally into the parenchyma of the pons, and form a small, well-defined motor nucleus. These neurons function in the general somatic efferent system and innervate the muscles of mastication derived from the somatomers in branchial arch 1. The sensory neurons in cranial nerve V are derived primarily from neural crest cells that form the trigeminal ganglion. Most of these neurons are axoneurons, and their dendritic zones are widely spread over the entire surface of the head and to the mucosa of the components of the respiratory and digestive systems located within the head. A smaller component comprises general proprioceptive neurons for the muscles and joints in the head region. These sensory neurons greatly outnumber the motor neurons. Therefore, when these sensory axons enter the alar plate region of the metencephalon, they spread out for a short distance rostrally and for a long distance caudally, forming the spinal tract of the trigeminal nerve in the pons and the medulla. These axons terminate in telodendriote synapses in the alar plate neurons, which form the sensory pontine nucleus of the trigeminal nerve in the pons and the nucleus of the spinal tract of the trigeminal nerve in the medulla. This spinal tract and nucleus extend the full length of the medulla, where caudally they meet the analogous functional neurons developing in the first cervical spinal nerves and spinal cord segment. See Figure 3-11. The cerebellum, or dorsal metencephalon, is formed primarily from the proliferation of the germinal epithelial cells of the alar plate, forming the rhombic lip. See Figure 3-11, Figure 3-12. This growth dorsolaterally from each side replaces the roof plate of the fourth ventricle so that the cerebellum forms part of the dorsal boundary of the fourth ventricle in the metencephalon. The development of the cerebellar cortex and nuclei is described in Chapter 13. The ventral metencephalon is the pons. A ventral migration of alar plate mantral layer neurons forms the pontine nucleus. See Figure 3-12. The axons of these neurons cross the midline and course dorsally into the cerebellum. This forms the transverse fibers of the pons, which demarcate the ventral surface of the pons and continue into the middle cerebellar peduncle. Mesencephalon, midbrain symmetric proliferation of the walls of the neural tube in the mesencephalon reduces the size of the neural canal to a narrow tube, the mesencephalic aqueduct. This is smaller rostrally, where it joins the third ventricle of the diencephalon, and larger caudally, where it is continuous with the fourth ventricle ventral to the rostral medullary velum. Cranial nerves 3 oculomotor, and 4 trochlear, are associated with the midbrain. These contain primarily axoneurons that innervate extraocular muscles. The cell bodies are in nuclei derived from the basal plate mantral layer. They do not migrate but remain adjacent to the median plane ventral to the aqueduct, which is in the same topographic nuclear column as the abducent and hypoglossal axonuclei in the medulla. Figure 3-13. The parasympathetic nucleus of the oculomotor nerve is just rostral to the oculomotor nucleus. This contains the neuronal cell bodies of the preganglionic parasympathetic innervation to the constrictor muscle of the iris. These are derived from the same basal plate mantral layer. The alar plate proliferates dorsally to form the tectum of the midbrain, which is divided into paired rostral and caudal colliculi, collectively known as the corpora quadrigemina. These are associated with visual and auditory reflex functions, respectively. The crus cerebri on the ventral surface of the midbrain results from the caudal growth of axons from telencephalic projection neuronal cell bodies. These are continuous from the internal capsule in the diencephalon. An abnormal accumulation of fluid dorsal to the corpora quadrigemina has been recognized in dogs and is referred to as a quadrigeminal cyst. However, no studies have confirmed the presence of an epithelial lining, which is required for a cyst. Some of this may be a diverticulum of the caudal part of the third ventricle or the rostral part of the fourth ventricle or a space lined by meninges. Until the lining of this fluid-filled space is defined histologically, supracollicular collection of fluid would be a better description. Diencephalon, interbrain rostral to the mesencephalon, the sulcus limitans is no longer evident in the neural tube, and the diencephalon and the telencephalon are considered developments of the alar plate. The symmetric development of the lateral walls of the neural tube in the diencephalon reduces the neural canal to a vertical slit on the median plane. The third ventricle, adhesion of the developing thalamus in the center forms the interthalamic adhesion and separates the third ventricle into a small relatively round dorsal component and a larger vertical narrow ventral component. These two portions converge caudally at the mesencephalic aqueducts and rostrally at the level of the interventricular foramina, which connect to each telencephalic lateral ventricle. Figure 3-14. On the dorsal median plane of the diencephalon, proliferation of the neural tube epithelial cells does not occur, which leaves a single cell-thick roof plate, where a small choroid plexus develops in two parallel lines similar to those in the medulla. At the interventricular foramina, each of these is continuous with the choroid plexus that develops from the roof plate in each lateral ventricle. Figure 3-15. In the diencephalon, a plethora of nuclei is formed from the mantle layer and dispersed diffusely through this brain segment, thus forming a complex of nuclei and neuronal processes. These nuclei form the thalamencephalon, the hypothalamus, and the subthalamus. The thalamencephalon consists of the thalamus, the metathalamus, and the epithalamus, comprising those nuclei located dorsal to the ventral portion of the third ventricle. The hypothalamus includes the nuclei located on the sides and floor of the ventral portion of the third ventricle. The subthalamic nuclei are located ventrolaterally in the diencephalon. A ventral outgrowth of the hypothalamus, including an extension of the third ventricle, forms the neurohypophysis and hypophyseal cavity, respectively. The neurohypophysis becomes associated with a dorsal extension of the adjacent oral ectoderm, the hypophyseal, Rathke, pouch, to form the hypophysis, pituitary gland. The optic vesicles that initially grew out of the prosencephalon will form the neural layer of the eye and optic nerves, which become associated with the diencephalon. See Figure 3-3. The axons that grow caudally from the retina and the optic nerve will form the optic tracts of the diencephalon, and many will terminate in a nuclear area of the thalamus. These optic nerves form cranial nervi, the special somatic afferent neurons of the visual system. By definition, a nerve is a collection of axons outside the CNS that are myelinated by Schwann cells, which arise from the neural crest. Optic nerves are misnamed because they develop as extensions of the prosencephalon. They form in the optic stalk that extends from the diencephalon to the optic cup. Their axons are myelinated by CNS oligodendroglial cells. This is important to remember because the optic nerves are affected by diseases that are specific to the CNS. Therefore, optic neuritis is a form of encephalitis. Conversely, polyneuritis does not affect the optic nerves. Telencephalon, cerebrum. The rostral boundary of the brainstem is the lamina terminalis of the diencephalon. It is the rostral boundary of the third ventricle. The optic chiasm is located at the ventral portion of this lamina, and the rostral commissure develops and remains in this lamina. It is at this level that the telencephalic vesicles grow out of the original prosencephalon, to form the two cerebral hemispheres that comprise the cerebrum. The lamina terminalis is located on the median plane between these two outgrowths. The telencephalic vesicles grow out of the prosencephalon a short distance rostrally and then in a large curve caudally and ventrally. The neural canal in each telencephalon is the lateral ventricle, which communicates with the diencephalic third ventricle via the interventricular foramen on each side of the lamina terminalis. See Figures 3-15 and 3-18. Each telencephalon forms a cerebral hemisphere. The two cerebral hemispheres form the single cerebrum. The hemispheres are connected by the corpus callosum. At one aspect of the medial wall of the telencephalic vesicle, the neuroepithelial layer of the neural tube does not proliferate and remains a single layer of cells that become ependymal cells comparable with the roof plate of the myencephalon dorsal to the fourth ventricle and the roof plate of the diencephalon dorsal to the third ventricle. As the rest of the telencephalon proliferates and differentiates, this telencephalic roof plate will be attached to the crus of the hippocampal fornix on one side and the stria terminalis on the other side. The choroid plexus of each lateral ventricle develops in this roof plate, as was described for the choroid plexus in the medulla. This is a curved structure similar to the structures to which it is attached, and it protrudes into the lumen of the lateral ventricle. At each interventricular foramen, each lateral ventricular choroid plexus is continuous with the choroid plexus of the third ventricle. An extensive development of projection axons occurs from diencephalic thalamic neurons to the cerebrum and telencephalic neurons to the brainstem. This gives rise to a thick layer of myelinated axons, white matter, between the diencephalon and telencephalon that is known as the internal capsule. Telencephalic neuronal cell bodies and white matter may be organized as described below. Cell bodies. The telencephalic neuronal cell bodies are located in one of two general locations. One is on the external surface of the entire telencephalon, forming the various layers of the cerebral cortex. The other is deep to the surface in subcortical basal nuclei. These are often incorrectly called basal ganglia. It must be kept in mind that ganglia are collections of neuronal cell bodies outside the CNS. Such collections inside the CNS are nuclei or cortices. Cortices are located superficially, and their neuronal cell bodies are in a continuous arrangement. Examples of basal nuclei are the caudate nucleus, globus pallidus, putamen, claustrum, and amygdaloid body. The cerebral cortex may be divided into 3 regions on the basis of evolutionary and anatomic features. 1. The archipallium. Pallium is a synonym for cortex, is the hippocampus, which is an internal gyrus, an area of cerebral cortex that has been rolled into the lateral ventricle and is not visible on the surface of the cerebrum. 2. The paleopallium is the olfactory system, which is composed of the olfactory bulbs, the peduncles, and the piriform lobe cortex. In animal evolution, these are the most primitive portions of the cerebrum. 3. The neopallium is a more recent evolutionary brain development and makes up the surface of all the gyri and sulci of the cerebrum. Figure 3-16. Comparative evolutionary studies show the continual development of the neopallium in higher animals, relegating the archipallium and paleopallium to a lesser portion of the cerebrum anatomically. The surface of the amphibian cerebrum is smooth, lacking any gyri. It is composed of the archipallium dorsally, the paleopallium laterally, and the basal nuclei ventrally. In the advanced reptile, the basal nuclei have receded from the ventral surface and have been replaced by the paleopallium on the ventral surface. A small lateral area is the neopallium, and the dorsal area is the archipallium. In mammals, the neopallium has overgrown the other divisions of the cerebral cortex so that the paleopallium is entirely on the ventral surface of the cerebrum, ventral to the rhinal sulcus, and the archipallium is rolled medially into the lateral ventricle as an internal gyrus, the hippocampus. Continual development of the neopallium in higher mammals has resulted in the characteristic gyri and sulci observed over most of the exposed surface of the cerebrum. The rhinal sulcus separates the neopallium from the paleopallium. This is characteristic of all of the domestic animal species, but most laboratory rodents and all birds have no gyri because in these animals the neopallium is unfolded, so the cerebrum has a smooth surface. Although at birth a few gyri and sulci are present in the puppy brain, they increase remarkably during the first 3 to 6 weeks of life. Axons. The axons of telencephalic neurons form 3 groups of processes on the basis of their destinations. 1. Association. 2. Projection. And 3. Commissural. The association axons course between cortical areas within one cerebral hemisphere. They may be short in course between adjacent gyri or may be long and traverse the entire cerebral hemisphere, but they never leave that hemisphere. Projection axons leave the cerebral hemisphere where their cell bodies are located and enter the brainstem via the internal capsule. They terminate in nuclei in various parts of the brainstem, with a few reaching the spinal cord. Commissural axons cross from one cerebrum to the other. Figure 3-17. All of these axons are intermixed in the white matter of each gyrus, which is the corona radiata, and in the centrum semiovale, which is the mass of white matter in the center of the hemisphere dorsal to the lateral ventricle. The semioval appearance of this structure may be appreciated only in a dorsal plane section of the cerebral hemisphere. All of the 3 groups of commissural axons initially develop in the lamina terminalis. Figure 3-18. The rostral commissure is located ventrally in the lamina terminalis and courses primarily between paleopial structures and basal nuclei. The amygdaloid body, on each side. This commissure remains at the site dorsal to the optic chiasm in the fully developed brain. Another small group of commissural axons courses between the archipallium, hippocampus, of each side. This commissure migrated quadodorsally as the telencephalon developed to reach a position dorsal to the caudal aspect of the diencephalon. The largest group of commissural axons forms the corpus callosum, which also expands dorsally as the cerebrum develops so that it is positioned between the other 2 commissures. The corpus callosum serves primarily to connect the neopial areas of each cerebral hemisphere. It begins in the lamina terminalis, and as the telencephalic vesicle expands, the corpus callosum enlarges and extends caudally dorsal to the diencephalon. Near the median plane, it is located between the neopial cingulate gyrus dorsally and the archipallial hippocampus and body of the fornix ventrally. Laterally, in each cerebral hemisphere, the corpus callosum forms the roof of the lateral ventricle. The septum pellucidum develops dorsally in the lamina terminalis between the genu of the corpus callosum and the rostral body of the fornix. See Figure 3-18. In the telencephalon, the neural tube germinal layer ultimately is replaced by the ependoma of the lateral ventricle, except for the area of the basal nuclei. The mantral and marginal layers reverse their positions. This is the result of the migration of the newly formed primitive neurons to the surface of the neural tube to form the cerebral cortex, where their axons grow centrally, forming the white matter on the inside of the gray matter. Radial astrocytes participate in this migration by guiding the neurons to the surface of the neural tube. Ultimately, 6 layers of neurons will populate the cortex internumbered from superficial, layer I, to deep, layer VI. The first neurons to migrate to the developing cerebral cortex will form layer VI. As more neurons arrive, they pass by those already there to form the rest of the layers in a reverse sequence. Thus, the last neurons to arrive form layer I. The basal nuclei are formed by neurons that migrate only a short distance from the mantle layer into the developing white matter so that they remain in a subcortical position. Remnants of the telencephalic germinal layer persist throughout the life of the animal, forming the subependymal layer, which consists of a variably sized population of small cells that are thought to function as stem cells and serve as a continuous source of glia and neurons throughout the life of the animal. Postnatal neurogenesis is now a well-recognized event, especially in the olfactory system and the hippocampus. This subependymal subventricular layer is thought to be the source of some of the glial neoplasms that arise in the brain. The development of the choroid plexuses of the lateral, third, and fourth ventricles is similar, but the adult morphology varies. The choroid plexuses of the lateral and third ventricle are small and form a thin, undulating veil that projects into the ventricles. The fourth ventricle choroid plexus is more robust and lobulated, especially where it projects through the lateral apertures. This is especially evident in horses and cattle. In addition, in older horses often an accumulation of cholesterol crystals, resulting from chronic bleeding, enlarges the choroid plexus. The accumulation of these crystals may present a sparkling gross appearance of the choroid plexus. Occasionally, in the choroid plexus of the lateral ventricle, this chronic bleeding results in such a large accumulation of cholesterol crystals and associated chronic inflammation that a mass develops and causes neurologic signs associated with increased intracranial pressure. This cholesterinic granuloma may be unilateral or bilateral, figures 3-19 and 3-20, and may be diagnosed by computed tomography, CT or magnetic resonance imaging, MRI. They may be very large when neurologic signs occur. The choroid plexus may give rise to choroid plexus papillomas and carcinomas. The latter may spread to other sites by way of CSF in the ventricular system in the subarachnoid space. Meningiomas arise from arachnoid cells and expand in the subarachnoid space on the surface of the brain. A common source is from arachnoid cap cells, which are found as a cluster of arachnoid cells on the apical surface of an arachnoid villus that projects into a venous sinus. These cap cells arise from neural crest cells. Meningiomas that bulge into the ventricular system arise from the arachnoid cells associated with the choroid plexus.